Bueno, buenas días a todos ustedes. Muchas gracias por acompañarnos aquí. Yo voy a hacer la presentación de esta conferencia. Esta conferencia lleva por título Soluciones de Imagenología y Lectores de Microplacas para Estudios Predictivos de Toxicología. Bueno, antes de comenzar, voy a dar agradecimientos al Cinvestav, al Laboratorio Centrales LANCE, al doctor José Tapia de Genética y a su servidor Daniel Talamás, que en conjunto organizamos la conferencia junto con las marcas Inolab, especialistas y Molecular Devices. El objetivo de esta conferencia es la difusión científica, cuya finalidad es divulgar la ciencia y el desarrollo tecnológico entre la comunidad de investigadores en México a través de esta conferencia, que imparte una de las expertas en la técnica, que es la doctora Platica Inolab. En la actualidad hay un interés creciente en el uso de ensayos basados en células más complejas para la predicción del desarrollo de fármacos y detección de compuestos tóxicos. En esta presentación se mostrarán datos multiparamétricos para modelos de cultivo 2D y 3D, utilizando soluciones automatizadas de confocal y de campo amplio. Los datos presentados incluyen efectos inhibitorios dependientes de la concentración de los compuestos sobre la complejidad de las redes tridimensionales. A su vez se discutirán los ensayos funcionales y morfológicos utilizados para evaluar organoides y esferoides tridimensionales que respaldan su uso en las pruebas de fármacos candidatos y para la evaluación de la toxicidad. Una semblanza de la doctora Platica Inolab. Ella es una científica para las aplicaciones de imagenología en Molecular Devices. Tiene un doctorado en biología en la Universidad de Virginia y una estancia de investigación postdoctoral en biología celular, donde ella utilizó imágenes de células vivas para estudiar la regulación del ciclo celular de mamíferos. Si me pueden con un breve aplauso para la doctora.
Hello, everyone. Thank you. Thank you for the opportunity to visit here today and to speak with you about imaging and microplate reader solutions for looking at toxicology studies. We appreciate the invitation and thank you very much. So I'll start by just giving you a little bit of an overview of the molecular devices solutions where we're really focused on advancing protein and cell biology. And what you can see here is different groups that I'm not going to touch on all of these today, but just to give you a little bit of a big picture of what our company is doing from the perspective of solutions for protein and cell biology. We have our microplate readers here, which I'll tell you a little bit about today. Our automated imaging solutions and our high throughput solutions, such as high throughput plate reader, wide field and confocal imager. Solutions for colony picking and cell line development, as well as automation solutions and electrophysiology. So a wide range of different solutions to look at protein and cell biology questions. And what I'm going to talk about today is focused on looking at how many of the researchers are moving from 2D into 3D space. And we'll talk about, in particular, looking at toxicology and looking at using 3D models, such as organoids, spheroids, as well as organ on a chip or microfluidic type of platforms. It's an exciting year for molecular devices. It's actually our 40-year anniversary. So I just wanted to start by letting you know we've been working on these innovative solutions for 40 years, starting back in 1983 with the founder really progressing from moving from the cuvette into the microwell plate with microplate readers. It goes all the way here to the end of last year, 2022. Molecular Devices has acquired a company called Celeste, which has a proprietary technology for patient-derived organoids. So coming, again, from the perspective of the instrumentation and the software, as well as the reagents I'll tell you about today, looking at 3D biology. Now, just to give you a big picture, I'm not going to focus have both single mode or multi-mode systems like you see here with our spectrum at 5-3x, where you could do absorbance, fluorescence, luminescence, all the way up to alpha screen, being able to even have an imaging cytometer. I'll show you some examples. And for assays, looking at viability, proliferation, cell health, and so on. The imaging family that I'll tell you about and show you many examples of looking at spheroids and organoids in predictive toxicology is the family of the Image Express. So we have a family of Image Express systems here. We have confocal imager, we have wide field imagers. For all of these, you'd be able to do live cell, fluorescence, and transmitted light. And I'll show you some examples of using this technology, for example, to do a cardiotoxicity live cell type of experiment or looking at neurotox. Now, let's take a step back and just talk about what do we mean by predictive toxicology? Well, there's been a lot of emphasis here on moving into cell-based assays for predictive toxicology. And some of the reasoning is for that, as you're aware, toxic compounds in the drug discovery cycle will cause them to fail in that process. And of course, having drugs get to market that then later turn out toxic is not only extremely damaging to human health, but of having then those drugs to start from beginning here to look for new compounds. So there's a lot of emphasis on cell-based assays or assays where you can much earlier in the discovery process determine if there's a toxic effect. In addition, there's been much emphasis in the last year and with changes, at least in the US with the FDA, where having a change here from moving from animal models into cell-based models. So this is all aligned here with a emphasis on doing this predictive toxicology in more cell-based and 3D models. And some of the toxicity areas that I'll talk about today include looking at cardiomyocytes, so stem cell-derived cardiomyocytes, which are able to recapitulate through the feeding and the activity. So using these in 2D and 3D, using things like brain organoids, 2D and 3D cultures of neurons to ask questions about neurotoxicity, liver or hepatotoxicity, and we'll look at how these are tying into looking at, for example, the toxicity of different anti-cancer compounds. So again, we're looking at predictive toxicology here because a large percentage of new drugs will fail in that process due to toxic events. So looking at toxicity here, we could break this into a couple of different areas. Here you see cell-based toxicity, looking at some cells here that are being examined for proliferation. So when we think of a cell-based toxicity assay, you might be thinking of viability or morphological changes. Do the compounds cause the cells to die? Do they cause them to change shape? How does that process work? Neurons, looking at, for example, using imaging process that I'll show you to study how the compounds might have an impact on neurons with regard to the outgrowth, the branching, and the neuronal health. We can look at changes in the cell organelles. So are you getting, for example, restructuring of the membrane? Are you getting internalization and so on? And functional assays, such as looking at calcium changes, we'll look at those in neurons for neurotoxicity as well as in cardiomyocytes for cardiotoxicity, and being able to then use those as a functional readout. I'm just going to introduce you to one of the reagents here. You can, with our systems, you can use reagents across the board. This is an example that I'm going to use in some of the assays to show you, so I thought it would be good to start with an orientation. This is the early tox cell integrity test. And the way this works is you have a dye. In this case, that dye for living cells is going to be red. And where the green is the dead. So these are using different intercalating dyes. And so depending on the cell health or the cell membrane will determine whether these are inside or outside in this case. So the dead cells you're going to see are green, and the live cells in this case are going to be red, where you can see that membrane is intact. So we'll look at an example first here. Using one of the microplate readers, a multicolor reader, the spectrum S93. And so in this case, you've got your cells from microplate using fluid cells, then being treated, treated with different compounds of activity. So again, red is going to be your live cells, and green is going to be your dead. Putting this in the microplate reader assay, and then examining within the software things like your EC50 and your dose response. And here you can see one of those results with steroid. So we were able to examine, for example, the live dead ratio and see that as determine the concentration here of the steroid, we are seeing an impact here on the viability of those cells. We can also, look a little bit further here by examining the live dead ratio as well as looking at different markers here. So here we're looking at an example of a luminescence assay, which is looking at reactive oxygen species, right? So these you can see DNA damage, you'll get lipid peroxidation, that hallmarks here of cell death, and cellular aging. And what you're looking at in the middle here is a result from two of our readers, our I3F, which is actually positioned here, I mentioned earlier, and a luminescence jet activity reader with spectrum. So what you're looking at here is the reactive oxygen RS levels in a luminescence assay. And here you're looking at similar, you can see similar effects here. Now this is a multi-spec with that early tox kit to see whether you are getting cell death when you're seeing a correlation there with the reactive oxygen species. It may be a little bit hard to see back there, but that is when you're able to identify, so you can do this luminescence assay along with the fluorescence assay to look at the viability. So this is one way that researchers might be using these systems to do some of that early cell-based predictive toxicology. Now we can look at the cells on that very same system. That system has an imaging cytometer. So you can do your absorbent fluorescence, luminescence, and so on, as well as looking at the cells. And you can then look to see, what we're looking at here is monitoring, in this case, the cells, the different cell types. So you can see a few different examples here. On the left are the cells, and on the right you see the identification. That purple overlay is the software identification of the individual cell, which means we can, for example, count and do a proliferation assay. We can monitor how the cells are growing. So we can monitor them on this microplate reader, as you saw by looking at luminescence for reactive oxygen species, look at live dead, and here we can visualize the cells as well. So we can see how treating with those different compounds might impact not just the cell number, but the cell shape, the cell health, all of these readouts here, determining if this treatment is causing a toxic effect. And you can see we can do this with fluorescence as well as with cells that are unstained. We call that stain-free. And what you're seeing here is a correlation to the comparison, looking at the cells, right? Either doing fluorescence or using just transmitted light, so unstained cells. And there's a very good correlation. So you don't have to, fix your cells and label them. You can also do this with unstained cells. So what I want to move into now is we're thinking about taking these assays from a 2D into a 3D environment with samples such as spheroids, organoids, and these cells grown in, for example, scaffolding type of matrix. And to do that, we're going to look at a few different examples within predictive toxicology, as I mentioned, cardiotoxicity, hepatotoxicity, and we'll look at neurotoxicity. We'll also look at within within cancer treatment, in particular leukemia, we'll look at some lung organoids and how the compounds might be impacting the lungs. So this is the general workflow where you have your cells plated. These could be in a multi-well plate. They can also be on slides. Here you're seeing the formation of a cancer spheroid here in transmitted light. This is in a round bottom well, so the spheroid's falling to the bottom. Staining those up with different markers, such as for viability or for mitochondrial integrity. And then imaging in an automated imager. In this case, we will look at some examples from confocal imaging as well as live. And what you can do is you can take two-dimensional images or three-dimensional. So we can take images through the spheroids or the organoids and look at a three-dimensional analysis, or we can look at a projection of those. When you do a 3D analysis, you're looking at things like volume or 3D cell count as compared to looking at a projection. So the first example, we're looking at screening of these tumor spheroids. These are colon cancer cells. They are being imaged every 3 hours in the image express system for 2 days. So temperature, CO2, and humidity, just like an incubator. And you can see over that time how the spheroid is forming at that bottom. This is the microwell plate. It's flipped over. So you can see that each of these is a little round well, and you're getting a spheroid right there at the bottom. So we can treat these spheroids with different compounds and look to see, again, how these compounds are impacting the spheroids with regard to morphology, the live cells, the dead cells, the cell shape, the cell expressions, and so on. And that's what you see here. This example is a 384 well plate. We have 6 different compounds, the anti-cancer compounds you see on the top here. We've got replicates, we've got dose response for each of them, and our controls on the bottom. So what you're seeing here are those spheroids where we have treated them, take the Z-staff, project them, and you're seeing that single image here. This is actually interactive, so you can click anywhere and look at those full resolution images. All the images are saved. And you can see very clearly, visually, between our control and our drug treated, we're having a big impact here in the size, the morphology of the spheroid. The color staining you see are the different markers for viability. So we want more than just pretty pictures, we want numbers. And how do we get those numbers is having software to be able to automatically find those different populations of cells and get the multi-parametric data. So you see on the top here the raw image, different concentrations of these compounds. And on the bottom, you're seeing the software mask for the identification of the different compartments. In this case, you've got your spheroids in blue, you've got live or dead cells in the different colors here. I'll show you coming up. So once those populations are identified, you can then make measurements. So we can see how, for example, a topocyte here is causing the spheroid really to kind of shrink up, as well as changing the number of live or dead cells. So these data can be output. We can look at our total cell number, how that changes in the spheroid cells, if you have markers for live or dead, like we did in this case, the area, and so on. In the next example, so this was actually an experiment where we took the Z-stack, we took the 3D data, but we collapsed it into a single image and analyzed that. What I'll show you coming up is taking the 3D data and analyzing, and then making a comparison and seeing, are there differences? So in the next example, here's the workflow. So we culture the spheroids for 4 days in those round bottom plates like you saw. Then we treat with different compounds, in this case for 24 hours. We use that same kit I mentioned earlier, the early tox kit, so that we can look at how many live cells and how many dead cells using fluorescent marker. We acquire those Z-stacks and then analyze in 3D. So again, looking at something like the volume or the 3D cell counts. And here you can see those results. So this is, again, looking at cancer spheroid toxicity. So at a high dose, and at a low dose, right? So again, red here in this case, sometimes you have to think about it because I always think green is live and depends on the kit. In this case, again, red is live and green is dead. So it's very clear. And if you look, this is a decreasing concentrations of camptothecin, so a topoisomerase inhibitor. And you can see that at the decreasing concentrations, right? If we look at our live and our dead cells, and then here you're seeing an overlay. So if we look at those in a little more detail, what you're seeing here, again, the image on the top, high dose, the images on the bottom, the low dose. We have different markers for live and dead populations here, right? Live and dead. And then the overlay. What's really nice about this kit is it's a one-step addition. There isn't a lot of washing. So you can, especially when working with 3D biology, with spheroids, organoids, they're kind of fragile. So you want to be able to reduce those washing steps when possible. So how do we look at 3 dimensions for the analysis? There's a couple ways to do this. The example that you're seeing here is we're looking at each plane. So this represents the Z step or how deep you're moving in to the 3D structure. And on the right, you're seeing the identification of the dead cells. The color coding here is so that it's easier for you to see two cells that are close together. So we can do that, again, at different positions throughout the object in Z. Here you see the live and dead overlay. So the color coding here is a mask. Again, in this case, yellow is live and blue is dead. And you see that overlaid with the spheroid. So we can then look at, as we look at decreasing concentrations of the compound, in this case, camptothecin, we can look at the absolute cell number, how many dead cells, the volume of the spheroid. Again, that's a 3-dimensional volume. So you can see that we could do this, and what you'll see in some of the upcoming studies is looking at a panel of different compounds and looking to see which ones have this topic effect. Now, I want to mention, moving now into some examples of organoids. Again, there's a lot of been a lot of advancements in spheroids, organoids, 3D biology from multiple areas, from the way that the spheroids and organoids are grown to the optimization of the formats, like the round bottom plates, or different types of formats to optimize for the 3D biology, the instrumentation, the software, all of this. So I want to kind of give you an overview here of that process for the organoids. So here on the left, you're seeing a 2D pre-culture. So in the 2D pre-culture, you have induced pluripotent stem cells that you are then growing here in 2D. Before you move them, they're going to develop into 3D organoids. Typically, this is going to be done, and it depends on the kind of organoid, but you're going to have a matrigel dome, so a 3D dome here, where you have the organoid forming, depending on the kind. Those organoids will grow depending on the kind of organoid. It could be 3 to 4, 8 to 10 weeks that they're growing, and then again treating them with different compounds and looking at different markers for either populations. Maybe you're looking to see, are they expressing the right protein for that particular organoid, and so on. And then confocal imaging them and looking at that 3D analysis. So in this first example, this was done with the Image Express Confocal HTAI. This is a laser-based confocal, a fully automated system. And what you're seeing here are some lung organoids. So we have epithelial cells that we're growing in culture and then transferring those into having a 3D culture. And what you'll see coming up is the growth of those and how these grow over several 6 weeks. So these, again, they're in the matrigel domes that I mentioned previously. And we'll use these lung organoids in the example to show you some of the toxicity assays. Here you can see 2 weeks up to 6 weeks. These are very large structures now, these lung organoids. And we then did some assays looking at, in this case, viability. So what you're seeing here is the image on the top. So in this case, we're looking at treating these lung organoids with TNF alpha and looking at live or dead. This particular assay is a 2D assay. So we're taking the Z-stats, we're projecting them, and then looking at the live or dead cells. in the organoid. On the bottom, you see the analysis. Again, the color here represents the software finding the individual cells. And you see here with the control versus the drug treated. On the right, you're looking at the percent in this case of live or dead. Right, so we can output that data, and what you're looking at the bottom is one example, which is steroid treated at different doses. And we can see that as that concentration increases, right, we are increasing the population here of the dead cells. So clearly this is having an impact on the whole organoid. As I mentioned, there's been emphasis here on trying to compare or to look in both 2D and 3D to see if there are differences. And that's what we're showing here in this example. So in this example, we're looking at, again, the images on top and the analysis on the bottom. And this is a compound, anti-leukemia compound. And the question here is this compound causing an impact on the lung organoids, on the lung cells at different concentrations? And on the right, what you're looking at in yellow is that 2D analysis. Okay, a 2D assay, so either in a flat culture or looking at the projection. And in black here, you see the 3D. And so what's really apparent here is there's quite a big difference. Exactly why in this particular assay there is that big difference, that's something further research is needed for. But it's an example of seeing that you can find differences in 2D versus 3D, and underscoring the need to look at both of those. The next example is going to look at some liver cells. So these are stem cell hepatocytes that you see here. These are hepatocyte cells from the urodynamics. Culturing these to form the spheroids. In this case, this is one of these ultra low attachment plates. And then again, treating with compounds, depending in this case, it's for three days, and then imaging them with confocal imaging. So we're looking at the nuclei, live cells, and dead cells with the Basidium diner albumin. And then we'll ask questions again. In this case, we were looking at a series of different compounds to see how they impact the liver cells. But things such as some known liver toxins, some things like flame retardants, and so on. So here you can see in that 3D assay control, darachlorine, ketoconazole, and halidorel, we can see that, again, they have different impacts here. We're looking again at the markers where we're looking at the population, in this case of live or dead cells. And we did this in the 3D analysis. So again, similar to what you saw earlier, but here I'm going to show you scoring as well. So this is looking at the nuclei count. Again, the colors represent, so it's easier for you visually to see the cells that are next to each other. And on the right, you're looking at scoring analysis. So this is the image, and this is the analysis map. And what you can see here, if you look at the legend on the right, basically what we're looking at is the cytoplasm and the nuclei of the live or dead cells, depending on these markers. And seeing how treating with these different compounds will impact these different populations. And here you can see an example with a known liver toxin here, methylmercury, compared to the control. Okay, so these are spheroids or liver microtissues. And then we can analyze that. Again, the legend here is what you're seeing here is a marked decrease in the number of live cells. In this case, they're yellow with this count. Right? You're seeing a large increase here of the number of dead nuclei. So we did this across a variety of different compounds. And here's what you can see, right? If we look at the dead cells, the volume of those spheroids, of those 3D tissues, or the diameter, and of course, depending on the compounds, we've got our control liver here. We've got something like mitomycin C at this concentration. We're not seeing an effect, but something like methylmercury, darachlorine, we're seeing very high. These are examples from liver tox, hepatotox. Let's take a step now to look at some live cells and look at cardio spheroids. So what you're seeing here are cardiomyocytes. So in 2D, the 2D culture that you're seeing here, then forming into the 3D spheroid. And we're looking at changes here. This is using a calcium marker, calcium dye. Like calcium dye from molecular devices, work with other calcium dyes. So you're seeing the transmitted light. Here you see that cardiospheroid. Cardioid has a couple different names. And this is a calcium flux assay. Right? We're going to see these events happen very quickly. Again, this is happening in an instrument that's doing this across the whole plate in an automated way. So you're just putting the plate in, coming back, and you've got these results. And then the intensity analysis is because we're going to look at, in this case, the whole spheroid, and we're going to look at how that intensity changes over time. And that's going to help us to understand how are these compounds, if they are toxic, are they causing the beat rate to change? Are they causing it to speed up, slow down, and so on? Okay, so you can see 3 examples here are control population. Isoproterenol, which is speeding up the beat rate, seeing much faster changes here with the calcium marker, and cisapride, which is slowing down. And here you can see an example where we're doing that in 3D over time. So control epinephrine, speeding up. And if you look at the bottom, you're looking at those spheroids over time, and this is over 2 seconds. So it's very fast. And you can see we have low, high, low, high, low, high, right? That's your beating. Using epinephrine, that speeds up. So the transitions from low to high, speeding up. And cisapride, slowing down. And we see that if we plot the intensity over time, is what you're looking at here. So in this case, we're looking at the intensity, that's in this axis, and then this is time. So control, you see rise, fall, rise, fall. And then if we look at some compounds like cisapride, which you just saw, slow down, that intensity changes, slow down, we see that it's slowed down. Some cause changes in the shape and so on. We can characterize these profiles by measuring things like the beats per minute, the amplitude, the spacing. So we can get a variety of different type of quantitative data about this beating pattern. Another example of doing a live cell toxicity assay is looking at neurons. In this case, I'm going to show you with Sarasorian treated first. We'll also then look in a 3D model at a microbrain. So let's take a look at this example, this 2D neurotox assay in unstained cells first. This is a Sarasorian dose response, so you can see time after plating. These are actually freshly plated stem cell derived neurons, so straight from the freezer into the plate, imaged overnight. Untreated, 10 micromolar Sarasorian, different time points, 0, 3, and 9. Now it's very clear, we can see that we don't have very nice, neurons with nice outgrowths and branching. But again, it's not just about what we can see with our eye, but the quantitative data. And so we do that by having tools to be able to find all the cell bodies and the outgrowths. So we can then look at a variety of different compounds and different concentrations over time and see, in this case with the Sarasorian, we're able to then, of course, measure that toxicity. These are 2D neurons in a plate. Here we look at an example of what's called a neurospheroid or a microbrain. This is from a company called Stemonics. They make these 3D microbrain ready assay plates. And what's really interesting about these is we can look at the fluorescence here to look at characterize the different cell types, but we can also look at activity because they will spontaneously fire as well. So let's take a little bit. So now you're seeing one of those neurospheroids. This is the 3D microbrain. So this comes from a single donor, a human pluripotent stem cell line. It has neurons and astrocytes, and you can see you've got the key markers here by the different stainings. And as I mentioned, what's really interesting about these is we get this spontaneous activity. So we can also look at the activity, and I'll show you that using one of our high throughput microplate readers and our imaging, and looking at both of those together. So let's look at the images first. You can see In this case, we're looking at, again, these are confocal images. This has been a clearing after the staining. And as I mentioned, these come as a plate with the micrograins in them. Okay, so staining for the different markers in this case, so the GFAP, MAP2, and the nuclei with GAPI. So let's look at the toxicity assay here. In this case, we see the images on the top. We've got, here's our methyl mercury again, by the way. We've got 3 different compounds on our control. And on the bottom is that mask. Again, that's the software showing you the spheroid in blue and the different cell populations in this case, in the green, orange, and the dark blue. Okay. So we can see, because we can identify these populations, we can then measure. As I mentioned, you have the activity as well. So what you're looking at here, if you follow this micrograin, you're seeing some spontaneous firing. So we're imaging this, but we can also put this on a high throughput microplate reader, our Clipper system, where we can then look at the calcium oscillations across the whole plate very fast. So you can see, we can measure. So again, this is looking at the spontaneous firing. So we're looking at that intensity as it changes over time in this case. And we can look at these profiles, so we can look at those traces, basically for different compounds across time. And what we see in these neuronal cells is here we can see, so what you're looking at here is the relative fluorescence over time. And you see the control. A couple of different profiles or different phenotypes that you might observe. Here's an example of GABA, where you've got a decreased frequency, right? So we're spacing those peaks out. Here you can see compound MK801 that causes variable amplitude. So you can see not a nice consistent pattern here. A lot of differences here in that profile. And you can see an example here with this compound where you've got not just decreased frequency, but also some irregularity in the spacing. So again, we can use these 2D and these 3D models in this predictive toxicology type of application to, really try to get at a lot of information here, detail here on the mechanism if you were screening unknown compounds. Let's look at another example of neurons in 3D. And this I'm going to show you is the first example of 2 where I'll talk about an organ on a chip plate. So these are this is a microwell plate. In this case, we're working with the Mimitas organoplate. It's an organ on a chip. This is the bottom. You're seeing the plate flipped over. So these are microfluidic channels. And this is a 3D neuron culture that you're seeing, and you're looking sort of face down on it now. We'll look at it from the side here coming up. Staining with different markers, and then imaging confocal. And then looking at, in 3D, the branching pattern and seeing how the different compounds impact that. Just to show you an example of one of the images, this is at 20x. And so depending on the assay, we're looking at different markers for mitochondrial integrity, for cell health as well. Some results here, you're seeing the projection. And on the bottom is that analysis map. So the red here is finding all the cell bodies and the outgrowth. So when I say projection, this is the way these the organoplate or these microfluidic chips work. You have a 3D structure forming between 2 pieces of glass in the channel. So, and you'll see that kind of turning around. What you're seeing here is that taking the Z-stack and looking at the projection. So again, what I'm showing you first is going to be a 2D analysis, then we'll look at the 3D. So control versus no treatment, we can see clearly, has a big impact here on that neuronal health. And here are some of those results. We can look at the outgrowth, the branching, the processes, how many cells were alive or dead, because we multiplex that with different markers. Okay, and so we do, see a decrease there, depending on the compounds, as an example of some of the compounds here. We also look at this in 3D space. This is what it looks like. Coloring here is the analysis, so you can see cell bodies that are close to each other separated. But we can look at that again in 3 dimensions. So now you're seeing a rotation, so you can get a little bit more feel of how those the neurons are growing between that glass. And we can look for those fibers, outgrowths, in 3D with the software tools. You can also look at the different markers, in this case for a calcium as well as mitochakra orange. So we can also output, where the cells are in this case in the structure with regard to vitally apoptosis. And that's what we see here. So this is that three-dimensional analysis, analyzing the whole volume. And looking at the fibers, the volume, live cells, dead cells. And what was found basically, again, there was a difference. If you look at, for example, the EC50 of those compounds, a difference between the 2D and the 3D assay. The next 3D assay I want to tell you about is a 3D angiogenesis or blood vessel formation assay. This is again in that organ on a chip environment. So you're going to actually get a 3D vessel tube formed to ask questions here about how the compounds impact the angiogenesis or angiogenesis processes. So this assay is often done with Quebec cells that are grown in a matrigel, and then you would look at a Z-stack and maybe do a collapse rejection. Here I'm going to show you doing this in 3D on one of those organs on a chip plate. So this is working with the Cinvestav organoplate. This is the microwell plate flipped over. You can see the chips here. So each one of this, in this case 3 by 3 is one. And we're looking again face on here. This is actually moving through the bottom here. So what's happening is these endothelial cells are forming a tube at the top. And at the bottom, there's angiogenesis factors. So over time, you're going to see this, which is that you get, and collagen is in the middle, that you get sprouting towards those angiogenesis factors. And we can measure that network of the amount of sprouting here. This shows you an example of, the raw image and how the software found the pattern and so on. That's what you're seeing here. This color overlay on the right is showing you how we can find this case it's like fibers, right? The network here as well as the individual cells. And so again, we can look over time. In this case, you're looking at over 4 days. And you can see, so what we get is a nice vessel forming here. And then over time, you get that sprouting towards those angiogenetic factors. And here they're being characterized by looking at actin and the pegyrin. And so we can look at the sprouts again in 3D, the volume, the nuclei, and see how that, over the course of days. And of course, if you are treating with different compounds, you would see how these change the volume as well as the number of nuclei in the structure. This kind of will show you, this is the tube at top. And we're looking at it from the side now. So we were looking at it kind of face on, now you're looking at it from the side. So you can see that's the tube. And growing down towards the factors, so that it's growing this way. This is looking on the face side, right? So the tube at the top. So this one's the tube and going down here. And this is how we actually measure it. This is one of the tools in our software called Express, where we're able to do 3D analysis. You've got your tubules forming on top. You're getting the growth and the sprouting through the ECM to those angiogenetic factors. The next example, in looking at different options for doing 3D biology, is looking at cancer spheroids in a micro pattern, in a micro well. So what you're looking at here, these are not different wells. This is actually one well, and this is the pattern. So it's like it has little kind of holes you can think about, where you've got different spheroids in each one of those. So this is looking at these cancer spheroids with different compound treatments. Again, looking in 3D to see how these different compounds are impacting the size, the shape, the volume, the number of live or dead cells, and so on. You can see here 2 different compounds. Go back here to our ferrosporin again. And you're looking at different dosages, right? So low you can see high, how the impact that you're having here on the population of live and dead cells, as well as on the integrity and the morphology of the spheroid in the microwell. So we looked at that control and then treated, again, what you're seeing here, the original image, and then the mask or the software identification. And then we can, for a variety of different compounds, we can see the diameter, the volume, again, of the 3D spheroid, and look at how these different compounds, right? You can see, for example, some of these do not have as much as a impact here, right? Versus some that are having, quite a big impact here in the dead cells. So I'm going to take a little bit of a pivot here and talk about the systems that I've been telling you about to do this automated imaging. Acquire the images, analyze, you can output the data, cell by cell data, or summary for the whole well, for the whole plate, so on. And then there's tools for doing the data mining or doing the data visualization, things like, doing that peak profile or doing mining here to find clusters and so on. So these systems really allow you to do something that, sitting on a microscope and doing the experiments that I just showed you is very labor intensive. I used to do this. I used to spend days and days in the dark room. The systems that I've been talking about today, the Image Express family, you put your sample in, slides in multi-well plates, you walk away, out could be on, it doesn't have to be in a dark room. Images will be automatically saved. You have a variety of different analysis tools, so you can really increase that ability to look at more compounds in your toxicity screening. You can look at more drug concentrations and so on. So you can save time as well as get a lot more data. And these are some of the different formats that are being used a lot in the 3D space. Just to kind of summarize, I told you about some of the microwell plates. So again, this is a pattern within the well. Round bottom plates, low attachment plates are very popular for working with the spheroids. This is a cross-linking gel. So these are looking at neurons in 3D space and a cross-linking. And then I showed you the example with angiogenesis and neurons in the Mimetos organoplate, the organ on a chip plate. This is a really interesting plate here. This one is called a graft plate. So what's actually happening is this tumor here in the middle is being vascularized so that the compounds that they're testing are being delivered to the tumor through that vascular network, right? Through that vascular network directly. The systems do enable you to work with stained fluorescence or unstained cells. Hold on, not going back. You see some nice examples here over time. Once it looks like it's catching up. I love watching these cardiomyocytes beat, so I have to let that play. Okay, well, if I had to have a technical difficulty, it's good when there's just a few slides left versus at the beginning. Okay, well, let's just let it rest for a minute, I guess. This is one of the Image Express systems. You can see transmitted light, phase contrast on top. You can do colorimetric if you want to look at tissues. There we go. Now it's all caught up. We've got a big preview there. Like I said, just a few more slides coming through. Love to watch these, right? So as you can imagine, you can watch this over time. We can see how, depending on the different compounds, you affect that beat rate, because you can look at the change in area. So you look at, we saw an example earlier looking at calcium fluorescence, but you can also look at how this structure changes area, and that will give you a readout there for the beat as well. Variety of different live cell examples. Again, the systems can have a full environmental control, temperature, CO2, humidity. Put your plate in, leave it in there for 2 days, imaging every minute, every 10 minutes, every hour, whatever for your experiment. This is an apoptosis assay, mitosis. You can see some macrophage engulfment and a wound healing or cell migration assay here. I want to just mention briefly, as we were talking about moving from 2D to 3D, one of the things to also think about is if you're doing this by an imaging approach, the images get more complex too. And something that's really, having a great impact in this area is machine learning. For being able to examine more complex type of phenotypes and look at more complex type of images. And so Encarta is our machine learning software that's used for segmentation and classification. So putting different phenotypes into different buckets. And here you can see an example of a few different, where we use machine learning, training different models, for example, to look at unstained cells, to look at images where you have a lot of variation in size of vacuoles in this case, looking at myotubes and so on. I want to end my next slide here is to show you how the tools that I've been talking about can actually be all tied together and automated. We have in our office in California what we call our Organoid Innovation Center, which is using our microplate readers, our imagers, and robotics in the middle, along with incubation and liquid handling. And I just want to play a little bit of this movie. We can send this to you as a follow-up. I'm going to play it without the sound. So you can see how, these processes can be all fully automated. I'll talk through it instead of the sound. This is my colleague showing you our office here. If you're ever in the Bay Area, we'd love to have you visit, so please let us know. And what you see here is our confocal system, a robot. You see our microplate reader back here. We've got liquid handling. There's an incubator in the back. There's an automated centrifuge. So literally, you could stack up your plates, and this could run those assays for you. Right, so you can use robotics with the microphones, which is our multi-mode reader I talked about earlier. Through the liquid handling, you can see both our imagers, our pico, and our confocal microplate washer. This is going into the imager. Back here, there's a little incubator, which you can't see, but it's an automated incubator. And yeah, this is highlighting specifically our confocal HDI, which is a laser-based confocal. You have water immersion objectives to allow you to go deep into the spheroids and organoids. So I'm not going to play the whole thing through. It's only actually a few minutes, but here you can see the optical design. It's a spinning disc confocal, which means you can do live cell confocal imaging for these organoids and spheroids. And then the 3D tools like the machine learning that I told you about, the 3D volumetric analysis. This is showing you an example straight from within the software for looking at these tools. You can see, you can find the cell borders, and you can find the organelles and so on. So with that, I will go ahead and thank you very much for, again, the opportunity, the invitation to be here today, and for your attention. And I'd be happy to answer any questions and would love to hear more about the research you're doing and hear more about your applications. Thank you so much. You want to take this? Okay.
To have a laboratory like the one that you showed, how much money do we need because we are in Mexico?
Well, it's modular, so you can build onto it. You could start with one, and then you can build on. All of those things are things that can be added later. So, and it's also modular from the perspective, it would depend on what instrumentation you wanted on your system. So, it will range based on that, but it is definitely something that you can add on. So I mostly present it from the perspective of, you can see sort of what options are available, what could, what potential there is.
But would you suggest like because to have all at the same time will be difficult.
Well, it's definitely something that, so we have a whole team. We have a great team. We call them our custom solutions or automation team. And we actually will want to know exactly the workflows that you want to do, the kinds of experiments, what you want to do now, what you want to do in the future. And this team of scientists and engineers, along with the applications, then would design that system. So it's not like one box. It's really designed. We have customers doing many different types of workflows and not kind of an automation one. This one we're highlighting specifically organoid and spheroid workflows. But it is something that sometimes people will get one instrument and then later as their throughput, as it gets more busy, as their assays change, they might want to add something to it, and that's possible.
Thank you.
Thank you.
Do you offer some training courses?
So thank you for the question. We offer training courses for the systems. Like if somebody buys an imaging system, there's a 2-day training we go through. We do the training with your assays and so on. We don't have a separate training course about imaging in general, for example. There are a lot of those courses, and some of those courses we participate in, but we don't host any of those courses ourselves.
Thank you.
Thank you. But we do have a full training.
By the way, very interesting. Everything.
Thank you. Thank you. It's
Beautiful
really an
images.
exciting time.
Very, very useful. I mean, it's clear that you get a lot of information.
Yeah.
in each experiment.
Yeah. Thank you.
Yeah, congratulations.
Thank you.
When you show a big difference in the results from the 2D and the 3D model, could you go further explaining that? I mean, does that mean that if we are working with 2D models, we are not getting the correct information?
No. I think it's just it's sort of, it's becoming more popular, the 3D, because the tools have become more accessible, like to ask those questions. I think for a lot of those questions, we don't really know. So we do some of the work in our office, like as part of our development, as part of our collaboration with customers. But a lot of the researchers doing this on their own that maybe would have more information about deeper into those mechanisms. So I think what we're sort of at this point where people are trying to now, it's becoming more accessible to do those 3D and ask those 3D questions and see if there's a difference. In some cases there is, in some cases there isn't, but it's more of a general, like having the capability now to have more options to ask those questions.
Thank you.
Yeah. Thank you.
Thank you. Well, when you're showing the orange and chip, can you do a co-culture of several different cell types? And they are all available from the same producer, I don't know, or you can pick them from different sources. I mean, if you want to do like a primary culture that you get from a patient, for example.
Right. Yeah, so the Mimitas organ on a chip company that we've done a lot of this collaborative work with, and they use our instrumentation as well. They have different disease models. So they have different chips and different formats for different disease models and protocols for those as well. But their customers can also be developing their own models using that microchip kind of environment to really be conducive to forming the 3D structures as well. So I think it depends on how you're setting that up, but they have a lot of experience with the different models that they work with and partnering with researchers on developing of other models. So it's hard for me to say an exact answer for that, but yeah, so this is, being able to, and those plates are also conducive for automation, but being able to have within that micro well plate, right, these, so that you can look at all the different treatments right within the same plate in that 3D environment. Thank you so much.
Bueno, buenas tardes. Antes que otra cosa, muchas gracias por su asistencia. De verdad, apreciamos mucho que se hayan tomado el tiempo para asistir a esta charla. De verdad, esperamos que haya sido de mucha utilidad, que salgan de aquí muchas ideas y muy buenas perspectivas en adelante para investigaciones futuras o las actuales que ya tengan, que se puedan complementar con información que se ha expuesto el día de hoy. Muchas gracias nuevamente por su asistencia. Este fue un evento realizado por Laboratorios Lance a través del doctor Tapia. Muchas gracias. Gracias al maestro Daniel Calamás por la organización de este evento. Muchas gracias a la doctora Penita Bormina por la plática interesante que nos compartió el día de hoy. Y nuevamente muchas gracias a ustedes. Por este lado, nada más para finalizar, y si alguno de ustedes tiene interés en acercarse a conocer uno de los equipos de los que pueden estar interviniendo en este tipo de estudios, pueden acercarse con todo gusto. Mi compañera Alejandra, que es analista de aplicaciones, les puede dar por ahí una breve charla y responder cualquier pregunta que tengan relacionada con esto. Muchas gracias a todos, que tengan excelente tarde y que estén muy bien. Hasta luego.